This podcast is supported by the Icahn School of Medicine at Mount Sinai, an international leader in research, education, and patient care. The school is the academic arm of the 7-hospital Mount Sinai Health System in New York City. It is consistently ranked among the top recipients of NIH funding. What are researchers in genomic medicine working on to transform patient care and improve patient health? Find out in a special supplement to Science Magazine, prepared by the Icahn School of Medicine at Mount Sinai in partnership with Science. Visit our website at www.science.org and search for Frontiers of Medical Research-Genomic Medicine. The Icahn School of Medicine at Mount Sinai. We find a way. Arizona State University and Science are recognizing researchers who address pressing problems. The ASU Science Prize for Transformational Impact honors early career scientists developing innovative solutions to today's biggest challenges. Hear from the inaugural winners on ASU's Lab Coat Optional podcast and visit science.org/ASU to apply. This is a Science Podcast for July 9th, 2026. I'm Sarah Crespi. First this week, researchers are protecting one owl species by hunting another. And contributing correspondent Warren Cornwall goes along for the ride. Next, we mark the 10th anniversary of the journal Science Immunology with its editor, Seth Thomas Scanlon. He takes us through the reviews published in the special issue on immune memory and what's next for the field. The northern spotted owl is kind of a symbol of environmentalism in the 1990s. Its endangered status put this owl's well-being in conflict with the logging industry in the western United States. And the owl won in court, putting a stop to the destruction of a lot of its wooded habitat. But another danger lurked for this small fowl. An invasive owl from the East Coast had entered its territory in recent decades and was outcompeting the native bird. The invasive owl, the barred owl's persistent growth has put conservationists in an odd spot. In order to protect the spotted owl, they are shooting the barred owl. Contributing correspondent Warren Cornwall went along on one of these owl hunts. Hi, Warren. Welcome back to the podcast.
Hi, Sarah. It's good to be here.
We have some good tape here from your trip.
We'll go ahead and start. And since we detected them earlier, I'm not going to start with spotted owl calls. I don't want to risk attracting any spotted owls that may be present.
I spent several nights out in the woods with Danny Hofstadter, who is a scientist from the University of Wisconsin at Madison. And he is leading this initiative that's a combination of research and I would say like wildlife management.
This is a really good example of an adult. You can see these very worn.
I'm with Danny. The way he spends many of his nights is he climbs into a big pickup and drives around on these remote dirt roads in the mountains of Northern California. They're, very steep, heavily wooded, and full of owls, mostly barred owls, but some spotted owls. He and the people that he's working with have created this incredibly complicated map of possible owl locations, and they're systematically visiting every single one that they can get to, trying to lure in barred owls to see if they're there.
So that's why we keep hearing the calls, like the noise of them?
Yeah, so the way you do it is dark is the best because it's when the owls are the most active. And so you go around to different locations and you play this recording over the speaker. And depending on the circumstances, you'll start with a recording of a spotted owl, and then you'll progress to the call of a barred owl. And if things get really exciting, they have all kinds of other recordings of like rodents in distress.
Oh, so food noises.
Yeah, exactly. The goal is to lure in these barred owls to within range, spotlight them with a flashlight so that you're sure it's a barred owl. And then if it is, you shoot it.
Yeah, so we don't have any tape of shooting, but we do have kind of the aftermath here where your host was basically picking out identifying marks on this barred owl. Here, I'm going to play that.
Before I bag her up, I just want to show again how I aged her as an adult. So we're looking at the terminal bands across the flight feather that last light colored band.
Yeah,
And
and
it's
again,
broad
we hear
across
lots
all
of
of
"Who
them. And
cooks
you
for
can also
you?"
see,
the barred owl call in the background. I think owls are very charismatic, so it's actually kind of hard to kinda get over the idea that you would have to kill one, but this is not that rare in the world of conservation. Getting to the kind of the science angle here, how did they decide that this was the way to go? Basically, finding all the owls and killing them. Why is that kind of the only option for protecting the native species from the invasive species?
Well, it's the only thing that's shown any results. These barred owls are incredibly well adapted to the environment. It sounds crude going out and just blasting away with a shotgun. But, what other option would there be, right?
They look similar, they're pretty similarly sized, they have overlap in their diet and where they live. And their territoriality, which is why they respond to these calls, is kind of the problem here.
But the spotted owl, until the barred owl showed up, was in some ways sort of the king of the forest from a bird perspective. I mean, it was a very effective predator. It sort of ruled the roost until the barred owl showed up.
They're really common on the East Coast.
Yeah, yeah. They're endemic to the East Coast. And even using the term invasive is controversial to describe them on the West Coast because it's not like somebody packaged up a box full of barred owls, drove them to California and set them loose, right? They got here under their own steam. It's not clear exactly how it happened, but one of the leading theories is that prairie land in Canada that had previously been a barrier to the owl began to fill in with trees once European settlers showed up and started planting trees and keeping First Nations from doing the burning that they traditionally did to keep the prairie open. It's possible also that a warming climate made boreal forests that had previously been off limits sort of open to barred owls.
We can blame humans either way. Either they built an owl highway
Yeah,
incidentally,
right.
or they warmed the planet so the owls kind of collided.
But they got to the West Coast under their own steam, as far as their own feathers, as far as we know.
Do we know how many of these endangered animals there are?
The short answer is no. The spotted owls have been incredibly well studied, but it's also a lot of work to count spotted owls, right? I mean, the traditional way that you do it is you go out into the woods and you hoot like an owl until it responds. There's no hard and fast number. Wildlife agencies have made estimates, but let's say in the ballpark of 8,000.
It's kind of living all the way from California up to Canada. Some of these conservation efforts are counting the mating pairs up and saying, we got this many. This is all we have. And other ones are like, okay, it's like 1,000. It's kind of varied depending on where you are. What do we have in the other corner here? How many barred owls do we think are in this territory?
More than 100,000.
Wow, that's a big ratio. So is this approach, shooting the owls, is that used now and across all of this land?
They're just beginning to use it at the management scale. It's really been experimental up to this point in these sort of small, localized areas because people didn't know if it would work or not. And, there's still open questions about, well, there's a lot of open questions, which is,
Does it work, Warren?
that's partly why Danny Hofstadter and his crew are roaming the Northern California woods shooting owls. There have been repeated experiments on the small scale that show that if people go in and start shooting every barred owl they can find, the spotted owl population will stabilize.
Okay, so stop dropping.
And when they compare it to similar neighboring areas where they don't do the shooting, the population of spotted owls continues to plummet. So pretty strong evidence that it's effective in that way. What they haven't seen yet on a significant scale is the spotted owl population starting to rebound.
But are we in a good time window for that? I mean, how long would that take?
And that's part of the question is these experiments have generally lasted about 5 years. There's one that's lasted a little bit longer than that. The thinking is these are not owls that reproduce quickly. It can take a while for a comeback, particularly if you don't have that many spotted owls left. So when you're starting with a small number, they're not going to be reproducing that quickly. And you also don't have many spotted owls in the surrounding area to sort of fill in the gaps.
Even if you do get that bounce back, you're going to want to look for all of these downstream effects of kind of rebalancing what's going on with the owls, right? You could see other creatures be affected by a change in who these predators are.
Yeah, and that's a big open question is what are the broader ecological effects? The biggest question mark around that is spotted owls are picky eaters. There's a handful of different rodents that are really their food of choice. Whereas barred owls, as one person told me, will basically eat anything that moves. When they have done DNA analysis of stomach contents in the barred owls that they're shooting. They've found, boy, north of 150 different species. And this runs the gamuts from salamanders to salmon to house cats, rodents, snakes, everything. The question then is, well, what's the ecological effect of having this top level predator that has such a wide ranging appetite? They just don't know.
What would be the goal or the target if this scaled up? How many owls do they need to kill?
The U.S. Fish and Wildlife Service, which is the federal agency in charge of recovering the northern spotted owl, issued a plan in 2024 for managing barred owls. And what that plan called for was creating these sort of refuge zones where they would be shooting barred owls in order to create safe spaces for spotted owls. And these zones were sort of throughout the range of the northern spotted owl in the United States. So this is all the way from the Canadian border down to really the suburbs of San Francisco. That plan, if implemented at its sort of most ambitious scale over 30 years, they estimated would require shooting 450,000 barred owls.
Wow.
That's a lot of barred owls.
That's a lot of owls. That's a lot of shooting. That's a lot of human time and energy.
That's the grand, most ambitious plan possible, as envisioned by the Fish and Wildlife Service. But on the ground right now, in most places, nothing is happening. Right now, you have a few state and federal agencies that are sort of tentatively dipping their toes in the water. So the Bureau of Land Management just last year started shooting barred owls in a few pockets of old growth forest in southern Oregon. And then you have really the most ambitious work is being done by Danny Hofstadter and this crew of researchers that he's working with in concert with two tribes that are in Northern California and a couple of private timber companies that own old growth forest in Northern California. They're really the big deal. The way I sort of am framing it is this is really the last stand of the northern spotted owl. When people look at Washington and significant parts of Oregon, the feeling is from a number of people that I spoke with is the odds of actually being able to get northern spotted owls back are low.
It's not necessarily recoverable there.
Yeah. I mean, there's one scientist who there's a study, it hasn't quite come out yet, but I got a copy of it that talks about how in a number of these areas, they're functionally extinct. You still have spotted owls there, but the numbers are so low.
Their population isn't, yeah.
Yeah, it's hard to envision them coming back. Whereas in Northern California, because it's the last range of the northern spotted owl that has been colonized by the barred owl, there's still enough spotted owls that people think there's still a chance. And so this is really the last stand.
Can you stop shooting?
Nobody could tell me a scenario where they could stop.
Oh, wow.
So this is probably forever.
That's some job security for
No, that's right. It's not that many people. It is a tough job. I'm telling you, it's no joke. The people that I talk to who are engaged in this are very committed to what they are doing, but they are not having fun. I think it takes a real toll. When I talked to Danny Hofstadter, I mean, he and other people who were involved in this didn't get into this because they wanted to shoot birds. They got into this field because they wanted to help birds. He, I think, has just decided that it is worth the price. And not everybody agrees. I would not say that there is a consensus that this is a great idea. I think that there's pretty broad agreement that if any intervention is going to be done that stands a chance of rescuing the spotted owl, this is it. That doesn't mean everybody thinks that you should do it.
Because between cost and kind of the ethics of it, I could see people being on either side. What happens with the owls that have been killed?
Right now, at least with the work that's going on in Northern California, they are stuffed into Ziploc bags, put in a freezer, and then eventually they're dissected at a lab in Northern California. And then various parts of the owl are shipped to Wisconsin, where they're stored for study. They're making use of the birds. I guess I can imagine that at some point you just have such a sheer volume of owls that you don't do much of anything with all of them, but that's what's being done at this point.
You said before it was kind of this like between management and research. Just as a scientist, it's got to be a strange place to end up.
I think it is, but I think in a way, Danny really feels like he's found his place. He decided not to get a PhD because he really wanted to have one foot in the research, but one foot out on the ground. That's his happy place. So I think he really feels like he is on the front lines. And boy, I talked to a number of people who basically said, we wouldn't be covering such an ambitious area. We would not be pursuing this at the scale that we are if it weren't for Danny. Danny is really sort of single-minded. I mean, I likened him to Captain Ahab in his relentless pursuit of his white whale, which in this case happens to be a small, fluffy bird.
This is such a stark contrast to Gretchen's story from, I guess it was a couple weeks ago, on the recovery of the wolves in Europe and how basically no one was allowed to shoot a wolf for several decades unless like a permit was issued for that specific wolf. And they've had such a comeback now that they're kind of everywhere and people want to shoot them because they're being intimidated.
Ecological puzzles are different, involving different management strategies. But as you noted earlier, I mean, the conservation world has a bloody streak to it, whether it's controlling invasive rodents on islands or shooting barred owls that are moving into territory of the northern spotted owl.
I can see that not everyone is going to agree this is the way to go, shooting barred owls to protect spotted owls. What are some of the arguments people are making against this?
The opponents have a number of arguments. The two leading arguments are this is unethical to do. You are sort of playing God between two different species of birds. And the other argument is just the logistics and the cost of this.
It's always going to go up, and the more you shoot, the harder they are to find.
A number of the people that are leading that argument are sort of animal welfare activists, understandably. I spoke with Eric Forsman, who was really a pioneer in northern spotted owl research. I mean, his work for the Forest Service over the decades really laid the groundwork for protections for that bird. He lives near a forest that used to have spotted owls in it in Oregon, and now it just has barred owls in it. He's not sort of actively speaking out against it, but if you ask him, he'll tell you that he thinks that there's just so many barred owls, particularly where he is, that he wishes the folks working in California luck, but he's watched this kind of wave of barred owls just fill up the forests.
We're not going to end on that.
Yeah, I mean, stay tuned, right? It's yet to be determined what happens. And, I mean, part of what will be interesting is, do state, federal agencies pick up this baton? Right now is being carried by scientists. This group of University of Wisconsin folks are operating on like 5-year grants. They're just sort of cobbling together this patchwork of money in order to pay for the work that they're doing. We're talking decades. I mean, this, as we discussed, this is going to have to kind of go on forever. Research like that does not go on forever. And so, one of the big questions beyond just the scientific questions of, can you do this at scale? How much does it cost to do spotted owls ever start to bounce back? One of the other questions is, are government agencies going to step in at a scale that people think is necessary or not?
If you have staff on all these different places that are working for conservation purposes, are they going to have to pick up a gun?
Stay tuned.
All right. Thanks, Warren. It's fascinating as always.
Good to talk to you, Sarah.
Warren Cornwall is a contributing correspondent for Science. You can find a link to the story we discussed at science.org/podcast.
Shipping, billing, admin, payroll, marketing. You're managing all the things. So why waste time sending important documents the old-fashioned way? Mail and ship when you want, how you want with Stamps.com. Print postage on demand 24/7 and schedule pickups from your office or home. Save up to 90% with automated rate shopping. That's why over 1 million small businesses trust Stamps.com. Go to Stamps.com and use code PODCAST to try Stamps.com risk-free for 60 days.
This is the sound of confidence. Confidence behind the wheel of the Mazda CX-50 Hybrid. It comes standard with a suite of proactive safety features and lets you explore far and wide with an EPA-estimated 38 miles per gallon combined and electronic all-wheel drive. Mazda, move and be moved. Tap the banner to discover how confidence comes standard. Mileage will vary based on vehicle condition, driving habits, and other factors. Used for comparison purposes only. Actual results will vary.
Also on the site this week, we have an online story by freelancer Hannah Richter about whether or not Uranus and Neptune are actually so-called ice giants. This story kind of makes you question everything you know about how to categorize planets. Worth a read. We also have a story from contributing correspondent Andrew Curry on just exactly how many indigenous people lived in Australia before contact with colonists. It turns out there were probably many, many more than have previously been estimated. And he also covers whether the first British ship to visit brought smallpox to the continent. In the commentary section, we have a book review by Wu on Cory Doctorow's new book, The Reverse Centaur's Guide to Life After AI. How to Think About Artificial Intelligence Before It's Too Late. Wu writes, quote, "This book argues that there's a potential future for useful AI, but not if we insist on subjugating rather than empowering human users." We see this debate coming up in the scientific literature a lot. Yes, AI, especially machine learning and other kinds of processing, can be really helpful for figuring out science, but do we want AI to do science for us and maybe not do it so well? It's really interesting to read the essays he's put out so far, anyway. From the journals, we have a science paper that caught my eye by Bao et al. Called Roots Navigate Around Decay Regions by Sensing Local pH Gradients. I like to see things that aren't supposed to be moving, and then learning how they navigate the world. It's a very cool idea. And in Advances, this was actually written up in the newsletter as well. There's a paper by Veach et al. On a debate about whether the marks made on the bones of an extinct elephant 50,000 years ago came from Homo florensis. This is a very short hominin that's extinct now, sometimes called the hobbits. So, were the marks on these extinct elephant bones 50,000 years ago made by Homo florensis or by Komodo dragons? This paper says, yeah, the hobbits were eating dragon scraps. They were eating what the Komodos left behind, according to these marks on the bone. Okay, back to the main show. We're going to be celebrating immune memory on the 10th anniversary of the journal Science Immunology. This week, we're celebrating the 10th anniversary of the journal Science Immunology. Seth Thomas Scanlon, the journal's editor, is here to talk about the first 10 years and what's next for the publication. Hi, Seth.
Hi, how's it going?
Good. It's snuck up on me, this anniversary.
It hasn't for me. I started now about almost 3 years ago, and it's sort of been in the sights ever since then.
Yeah, the podcast is coming up on its 20th anniversary.
Oh, wow.
You know?
So maybe I should interview you in a few years.
Yeah, at the end of the year. That sounds good. So looking back over 10 years, what stands out to you as some trends that you've seen, over the life of the journal?
A lot of the things that we initially identified as really important. So this was the very first issue. Angela Colmone, my sort of predecessor, they sort of identified areas that were really important. One was that tissue-specific immunity was going to be a really important organizing principle. So, for example, a few years ago, we showed how T helper cells that are residing in the lung are supporting sort of the optimal development of resident memory B and CD8 T cells in the context of flu infection. So that's one area. Or how a few years later, we had a paper that looked at macrophages. Immunologists love sort of subsetting these cells into
Finer and finer categories.
finer and finer categories, yes. And so they found that there were 3 distinct subsets that are conserved in the tissue that are really important. So that was one area that I think is really important and continues to be really important. Another is that I guess that human immunology was going to be sort of at the cusp, because now you have, I think, one increase in sort of technologies that are allowing us to explore these cells at higher and higher resolution. And I think the world is getting ever more connected. So if you actually look at these studies, they're often by multiple universities in multiple countries. And one, I would say, third thing that they identified back then, which has really borne a lot of fruit, was that the treatment of various disorders, cancer, autoimmunity, it was going to be sort of transformed by advances in immunotherapy and related technologies.
We're definitely seeing that happen.
Clearly happening. There's really great data out, for example, on the treatment of pancreatic cancer, not published in Science on Immunology, unfortunately. But and the other big thing was, of course, RNA technology, which
Oh, absolutely.
none of us could have anticipated was going to be so important.
I'm going to ask you at the end what to look forward to 10 years from now. But for now, we're going to talk about the special issue that kind of came out with the anniversary. The 10-year anniversary special issue focuses on memory in the immune system, which took me a minute to oh, yeah, well, we're looking back. We have memories. So very cool, almost pun-like selection. Can you talk about what went into the special issue? You got 3 reviews here. What are kind of the different takes they have on immune memory?
The first one is from 2 researchers at McGill University, Maziar Divangahi and Eva Kaufmann. And that's looking at the sort of evolution of the innate immune memory. So to give you a backup for your listeners, the immune system can largely be divided into this more ancient, quick reacting innate arm, which in theory reacts the same way every time, and the adaptive, which is what changes. So when most people talk about immune memory, they're thinking the adaptive because you see a pathogen once, and then the immune system is able to adapt to that and respond more quickly.
That's our vaccine strategy in one.
Right. But there's this whole body of evidence looking at this concept of what's called either innate immune memory or trained immunity. And the idea is that the innate immune system too can be kind of modulated by experiences that the host has. With certain more primitive organisms, this actually can be sort of a permanent change that will then last for a few generations and then will either be maintained, or because it's actually metabolically costly to be kind of primed and ready all the time, will eventually be lost. So this first review is really nice. It's looking at trained immunity, but kind of in the context of evolution and looking at sort of the evolution of early organisms and where sort of this concept of trained immunity came from.
And we have it in people too.
Right.
We have this inherited immunity.
And the point of the review is also that it's not just cells of the innate immune system, like macrophages and neutrophils, but also things like hematopoietic stem progenitor cells, HSPCs, which become all the various flavors of immune cell. These can be changed as well. And structural cells, so like endothelial cells, fibroblasts, and epithelial cells, which I would say immunologists are claiming now to be part of the immune system. We're taking more and more territory.
No. No more cells.
So a lot of this comes down to epigenetic changes. And if I were to say there's one theme for the whole special issue, is that these sort of epigenetic changes are really important for both innate and adaptive immune memory. And very nicely, this review looks kind of then at the role of this sort of innate immune memory in human lifespan. So early life, what are the things that are happening? For example, antibodies that come from the mother that then modulate permanently epigenetically change macrophages.
And then that can go away as you get older, or you can have more changes accumulate.
Or it can have permanent effects if you've been breastfed or not been breastfed, or the microbiome that you interact with in early life. Often there are these defined windows of time. And if you don't have that interaction, you don't see certain microbial metabolites. It's important to state that this kind of epigenetic memory can be both positive. It can, for example, enhance vaccine responses, or it can be negative. For example, when they talk about late life, this whole thing of immunosenescence, inflammaging, a lot of it comes down to your innate immune cells, they're responding too much.
That's super interesting.
So that's the first, this kind of setting the scene. The second review is from Stephen Turner, Danielle Thiele, and Nicola Gruda. All three are Monash. And that's looking at sort of the evolving understanding of T cell memory. So T cells are these key players in the adaptive immune system. It's a really nice kind of look at sort of the history and how the changes and advances in technology have really opened up our understanding of, in this case, this phenomenon of T cell memory. The key argument is that T cell memory, I mean, we have all these defined subsets again, very popular with immunologists, so central memory and effector memory and tissue resident memory. But I guess the understanding that's developing and what this review is highlighting is that T cell memory is a dynamic continuum of states, and it's actively maintained epigenetically, and that it's understood as flexible, a regulated fate rather than a static endpoint. They talk about future directions in this field. For example, a lot of this work has been done so far with the specific pathogen-free mice. So they have a limited number of pathogens. There are, for example, attempts to make mouse models that are more reflective of the human immune system. They have wildling mice, which we published when I was at Science, or pet store mice,
Yes.
dirty mice. And so that's one way that some of these important seminal experiments could be repeated now with these mice with more complex immune systems. The other thing that is a question near and dear to my heart is how repeated antigen exposures and how exposures to multiple pathogens at once get integrated. Because in immunology, a lot of studies, it tends to be primary and secondary, and that's it. Whereas, for example, with flu, you're seeing it every year, and that's really important.
So that's the second review. So for the third one, we're going to switch over to B cells.
Yes. So this is now the other sort of major component of the adaptive immune system, B cells. So this comes from Mauro Gaia, ex Marseille, and Kim Good Jacobson at Monash. And it's looking at how memory B cells, how they develop, how they regulate. And there's a focus here on these tissue resident memory B cells. And so they look at things like what is known now about regulators that push B cells to becoming memory B cells. A lot of the research, at least, is on the lung, sort of lung resident memory B cells. And these cells are really important because they produce the antibodies that are important for long lasting protective humoral immunity, either from infection or from vaccination. So they're really critical cells. So some of the interesting little things in this review, one of the aspects that it focuses is how the spatial organization of memory B cells is important, and it can shape how they recall. And so, for example, the anatomical site of where you get a booster may be important
What?
because maybe you want to hit the lymph nodes that are near the tissue that you're trying to protect. The other aspect that's related, and I think it's important, is intranasal vaccination.
It's a big push to do this, right?
Yeah. So, for example, there was an approach called Prime and Spike that was developed by Akiko Iwasaki at Yale, where you can take the vaccine and then you follow it by intranasal spike booster. This is the COVID vaccine, and you can get a really strong BRM lung cell and tissue resident responses. It's a really nice review. I think the three of them together, you can't hit everything.
It's on theme, right?
It's on theme.
So that's a very good state of the play right now. We had a look back at the past 10 years. What is your opinion on where things are going to go in the next 10 years? Where do you see as kind of the growth areas people are going to keep expanding on?
There are certain areas that I think that science immunology is doing very well in terms of publishing. When I was first sort of interviewing to become an editor, it's almost 7 years ago, some of these were sort of hot topics and there continue to be. So things like inflammatory cell death and innate immune signaling. Now you're getting these small molecule drugs that are targeting these pathways that could be really important in the clinic. I think immune aging in general is a very hot area. I went to a conference a few months ago. A big focus was the matrix, the immunomatrix, the sort of interaction between the immune system and the structural cells, so endothelial cells, fibroblasts, and how these cells are talking to one another. So I think it's an area of big interest. And then I think finally, the sort of rise of next generation vaccine platforms that can target diseases, along with, for example, rise in immunotherapies with CAR T cells. So some of these things were alluded to 10 years ago. They're kind of ongoing, but they remain of high interest.
It is so interesting as someone who's really only dipped into the field every few years, to hear all of these new kind of directions. Like epigenetics and immunology was not on my radar in the 90s at all. It is so interesting to hear how integral it is at this point. Is there anything we didn't talk about from the special issue that you wanted to touch on?
I guess a big part of the editorial is also just me thanking people. So I'm not going to thank them all again. I am just a very small part, and the history of the journal precedes me and will hopefully go on long after I'm dust in the wind. So I think it's really important to acknowledge the army of people that are needed to run any journal successfully. I think the people in production, art design, press pack, there are so many people that are involved in making the journal successful and making the impact of the papers as high as it can be. So I just want to emphasize how grateful I am to everyone and thank them.
All right. Thank you, Seth. Seth Thomas Scanlon is the editor for Science Immunology. We'll link to the papers, the reviews from the special issue, and also the cover at science.org/podcast. That concludes this edition of the Science Podcast. If you have any comments or suggestions, write to us at sciencepodcast@aaas.org. To find us on podcasting apps, search for Science Magazine on YouTube or Science Magazine Podcast. As always, you can listen on our website, science.org/podcast. This show was edited by me, Sarah Crespi, Megan Cantwell, and Kevin McLean. We had production help from Podigy. Our music is by Jeffrey Cook and Wen Koi Wen. On behalf of Science and its publisher, AAAS, thanks for joining us.
Have no fear, Chosen Foods is here to defend your favorite foods from the forces of seedy oils and sketchy ingredients. With cooking oils, salad dressings, and mayo, all powered by the good fats from 100% pure avocado oil and simple, delicious ingredients. Chosen Foods.